This is the session essentially on genome diversity network, which is essentially what we're trying to look at is ancestral diversity. And so this network is forming, and we welcome people to join, whoever is interested. What we really want to do is to build a cell atlas of all humans, and be as much embracive as possible. And that's because diversity is something that we celebrate. Diversity is a scientific frontier, and therefore we would not like to actually exclude and be inclusive of all humans, the entire humankind, the entire humanity. So that's essentially the purpose of the Human Cell Atlas network. The aims are, of course, to characterize various cell types, cell states, etc., activation of individual loci across a diversity of human populations. And of course, in looking at this diversity, we need to look at the kind of diversity in various, with respect to various covariates, gender, geographical place of residence, the environment that they're exposed to, the time, meaning perhaps age or physical time, the lifestyle inclusive of diet, and also whatever we have learned from genetic studies on these individuals from various populations. We use eQTLs and other QTLs to interpret the GWAS findings and identify mechanisms of known pathogenic variants. And you've heard talks this morning from Tuli and others, where all of these kinds of things are being done, and to be able to interpret properly the GWAS findings, we need to be embracive of all of humanity. Wish to harmonize and share data that's given in the Human Cell Atlas, that we would share data and in a harmonized fashion, and also the metadata that we will generate, that all of these, the data sets that we generate will come with a lot of metadata. We would like to share the metadata and harmonize all of that. Of course, this will require development of new algorithms, development of new pipelines for analyzing the kind of data that we will generate. And of course, we would expect the entire HCA community to contribute to the development of algorithms and pipelines. The healthy data that are being generated in all of this endeavor will actually provide us with insights into how to interpret and analyze data of relevant diseases that are of local importance or high prevalence locally. So this is what the HGTN aims are, and I would invite Shyam Prabhakar, who unfortunately could not be present in person, but he's standing by online, and he will present a few more slides. Shyam.
Thank you, Partha. Thanks for introducing the goals, and thanks to everyone for joining this session. If you're here, I presume you're already motivated and interested in the topic of human diversity. So we won't spend too much time on why this is important and why there are benefits to in all respects, right? Benefits to scientists, benefits to researchers, benefits to the communities, and benefits to medicine from having a diverse atlas. So we're going to jump in, roll up our sleeves here, and make this a fairly practical workshop type session. And the objective of this is to recruit you and your colleagues into joining the Human Genetic Diversity Network. Okay, not even that, even more concrete, right? Is contribute your data sets to diversify the Human Cell Atlas and to be a part of the Human Diversity Atlas version 1.0. Okay, so if you know the innards of how the HCA works, the wave 1 is well underway. There are 6 Human Cell Atlas bionetworks, including the lung atlas, for example, who are building organ-specific atlases. Wave 2 is just starting now. You can say this general meeting is the kickoff point for Wave 2. And the Genetic Diversity Network is in Wave 2. And we want to, so there's some concrete goals, right? To, if you want to be a part of this effort. And before I get to that, I say, what does it mean to be part of this effort? You all have your own projects, you have your own data sets that you will doubtless be analyzing and publishing on your own. Our goal is not to preempt that or to interfere with that in any way. Our goal is to do some specific meta-analyses across all your human cell atlas data sets, all qualifying human cell atlas data sets, to answer those very specific scientific questions that Partha laid out in the previous slide. Okay. So if you want to contribute, and we hope you will, you want to contribute your data sets to this integrative data analysis effort across the entire human cell atlas, there are two key deadlines here. One is end of this month. Please submit your project details. Just fill up one row of this spreadsheet. Okay, the QR code can take you to the URL of that spreadsheet and sign up. Like the key thing is put in the name of the contact person and the email address of the contact person. They put in the name of someone who will actually answer emails and join meetings. And then we'll follow up with that person to find out more about your data set and see how we can integrate it and include it in this atlas. Okay. And then the next deadline, if you have not already done so, is 31st August. Submit your data and metadata to Lattice. Again, we can point you to the right people. If you don't know what that means, just email us. The email address is up above. And I realize if you haven't gone down this path before, it could take some hand holding. So that's why step one is put a row in that spreadsheet and email us saying, I want my data set to be part of the atlas, right? If you want to understand the broader goals of the genetic diversity network, you can go to the second QR code. That's the roadmap for the Human Cell Atlas Genetic Diversity Network. It'll give you in much more details who we are, why we exist, what we're trying to achieve, and some way general outline. Okay, so this is the what we have as of August 2022 actually, but it has not changed much. The public PBMC, and we're starting with PBMCs. Some of you might have heard the session earlier this morning. We're starting with PBMCs because it's a tissue that has the largest number of data sets in the Human Cell Atlas and the largest cohorts, right? You need a decent cohort size to understand how sex influences our phenotypes, how age influences our phenotypes, how ancestry influences our phenotypes, QTLs and all that. So PBMCs will be the heart of version 1 of the Human Diversity Atlas. We are not ruling out including 1 or 2 additional organs, okay, that have sufficient cohort sizes and that have enthusiastic, motivated people who want to join. That would be fantastic. And we could, for example, analyze PBMCs in relation to tissue resident immune cells, for example, right? You can see here that the data sets are heavily European biased. On the left, you see the world map of countries, and the Human Diversity Atlas is very broad, but most of the data sets that have come in so far come in from Europe and North America. So we really hope more data sets will come in that diversify the atlas. And next slide, please. Okay, very broadly, what are the kinds of analyses we would do integratively with your data? Okay, so we would look at 4 major variables: the genotype, so QTL type analysis, ancestry, and ancestry is a loaded word in some ways. It's not easy to define, but it's one of those things where we all have a notion of what it means. But we're talking about genetic ancestry here, okay? And then sex and age. These 4 things, how do they influence your cell type proportions, your cell subtype proportions, your cell state enrichment, which a cell state, you can think of it as a subtype. And then how, what are the differentially expressed genes across genotypes, across ancestries, across male, female, and so on. What is the regulatory variation, meaning which transcription factors or chromatin modifying factors do we think are driving a lot of this gene expression variation? And then what are the QTLs, splicing QTLs, the expression QTLs, maybe chromatin QTLs, and so on. So this is just a bare bones skeleton of the kind of analysis we'll do integratively across all of the data sets in the Human Diversity Atlas. Okay, and if you have any questions, we can take them up as soon as Sophia outlines the discussion points for today.
Between Shyam and I, we have outlined what this particular network intends to do. There are many points to discuss, and invite Sophia to.
Okay, so we came up with a few discussion points, and we've broken them up into like general discussion points and methodology. So, of course, which data sets are ready for inclusion of the version 1.0 of the Human Diversity Atlas, what donor type, donor samples, protocols, and data quality descriptors can we include? How should we describe genetic ancestry, ethnicity, race, and other forms of identity? And which genotype profiling assay could we use or should we use? And then self-reported race and identity versus data-derived descriptors, discrete categories versus continuous. And in terms of methodology, how to associate genetic variants and or donor and sample metadata with single cell phenotypes, how to address batch effects and lab-specific technical variation and biases integrating the data sets, and then how to measure the degree of diversity in the HCA and how can we leverage insights from past population studies to inform the atlas. So these are all discussion points that are open, and we are ready to hear your thoughts, questions. Yes. Oh, hold on. Let's get it.
I thought that would be loud enough.
No, but we want to make sure that the folks online here.
Sure. Thank you so much. Very, very important. So my first point is related to number 1 point here, which is the data sets that it can be ready for inclusion. My question is, are you guys opening for including biobanks? Like, I'm asking this because we do have a very rich biobank that it has so much PBMC cells as related for each participant, plus other genomic whole genome sequencing data, and that's very population specific, and that's in Qatar. So that's, I think, not represented anywhere, actually underrepresented everywhere as a Middle Eastern population. I can help with that, and if this is something that I can add value to the atlas.
So is it already single cell data or
whole genome sequence data? We do have single cells. This is disease specific, which is not a biobank for the whole country. It is based on groups of people who work on certain diseases. So,
Shyam, you want to take that on?
So great question. Actually, if you could identify yourself, your name and institution, just for the audience. Yeah,
that's true. Sorry, my name is Amira. I'm a principal investigator at Cedra Medicine in Qatar, and yeah, I'm a part of the Human Genome projects of Qatar Biobank, and yeah. Thank you.
Thanks very much. Yeah, I've heard a lot about Sidra and the work going on there. Very exciting. At this point, for version 1.0, at least we're looking for single cell data sets that are already available, that are ready to go, right? So if you have, okay, there's some background noise. If people can mute themselves on the Zoom, or if the moderator can mute them. Yeah. Okay. Great.
Hang on, we can't identify the source of the noise.
It's Mary. But I can't mute. From here.
Just go ahead. We hope that it
Okay.
is reduced.
Okay. Thanks, guys. So right now we're looking not for whole genome sequencing data, although whole genome sequencing data will be very useful. We're looking for single cell RNA-seq data. So I should have made that clear upfront. We're looking for data sets where you have single cell RNA-seq data from a reasonably sized cohort. What does reasonably sized mean? ideally 100 or more, but if it's 50, 30 or something like that, we can still learn something. And then we'll add other data modalities, attack-seq, maybe proteome profiling, and so on. But right now, single-cell RNA-seq, primarily PBMCs, but other organs are also welcome. I hope
Shyam,
that
can I add on something to her question?
Please, yeah.
Since you're building this biobank, I know about the Qatar biobank and it's still being constructed. More and more samples are being replenished. If you wish to do a single cell sequencing, you must pay attention to the kind of protocol by which these samples must be preserved. Otherwise, these biobank samples cannot be profitably used for single cell sequencing. So if at a point of time you decide that single cell, you would like to join this consortium and network, then please, as you're collecting more samples, pay attention to the protocol by which you can actually do the single cell sequencing. Otherwise, you won't use the protocol.
Thank you very much. Of course, but this point exactly, it will be for a cohort, specific cohorts, not within the population biobank. It's like I have a cohort, okay, my own for type 1 diabetes, okay, children and adults. Those will go for single cell sequencing. Those right now I have around 500 individuals. This is something that I can contribute to because it's in the way, it is deserved the way that it should be. And we use 10x genomics for that. So but not full set as the number that you want. Let me finish it and I can just be in touch. I can register the project if it is something useful and can be done now until it's ready 100% with all the data available, then we can just start sharing.
So as Shyam mentioned, you can first just register. So go to both Google data, the QR codes. I will share that again, right? And put in your information. Since you already begun, you should also indicate whether or not you're willing to share what you have. If it's the PBMCs that you already have data on, maybe not 500, but yeah, going to add.
I will manage. Thank you.
Harry?
1.0, it's primarily healthy individuals.
Yeah.
For the biobank, yes, healthy individuals.
For what you are single cell, just single cell.
It's mostly, you said type 1 and type 2 diabetics, right?
Type 1, but also healthy controls.
Okay, so you can, the control data will be very
because
useful.
all our studies is control studies. So yes.
Well, Shari and Harry.
Hi, I'm Hari Nakshatri from Indiana University School of Medicine. I have 2 comments. One is regarding the genetic ancestry. Many of us have trouble convincing the reviewers that you have done the genetic ancestry analysis correctly. I've been struggling almost for the last 6 months with one reviewer in nature communication who would not, no matter how we do it, always, no, I don't. Particularly the admixture population.
Yes.
How do you name it? Right. So I think something that has to be, we need to be mindful about it when we frame that question or when we forward. So I don't know, Sam, whether you have a better answer to that. And my second point is in any of the GWAS studies or anything that we need when you're trying to put it together, you need to consider sex into consideration and how to separate them out based on the flurry of papers that came out recently, how the genes in the Y chromosome are influencing almost everything on the planet. Right. So that is something I did not see it in the write-up. That's all. And third is I already gave the indication of our data in your spreadsheet.
Right. I can answer the comments on the genetic ancestry a little bit. So are you, or asking me questions actually, are you using the single cell seq data and then, or is it independent? And they're giving you.
We are doing the genetic ancestry analysis independent of the single cell data using the DNA from the blood from the same donor. Right. So
That's
sorry,
one of the
Hari, if I can just jump in. The audio isn't great. You're doing the independently of what?
The genetic ancestry analysis, what we did of each samples is not using the single cell data, but rather the genomic analysis of the DNA from their blood.
I see. And the reviewer doesn't like that?
Yeah. No. The paper is not on single cell, but in general, whatever the method that you use to classify the people based on genetic ancestry, the reviewers have disagreement.
Are you classifying or just using bases?
I am not the reviewer, but I think the point is that any effort to try and put classifiers around genetic ancestry is, it's convenient. What we all do. We all know it's wrong. It doesn't make any scientific sense. It probably doesn't make, it reinforces the idea that these continental distinctions between ancestries are a viable way of classifying humans, which is clearly not the case. So we really don't have a solution for this at the moment, but I think if you're going to do this, you should collect enough data to allow you to future-proof yourself from all the evolution and the methods of quantifying ancestry in a more of a multi-dimensional spectrum, quantitative aspect. In the meantime, it may be convenient to categorize it into different continental groups, but as long as we understand that that is really just a convenience, not something that really represents the true state of nature.
Yeah, if I can jump in, that's a great point, Hari. And specifically, I guess everyone does their, self-reported ancestry or race or ethnicity descriptors, and then some genetic descriptors that are kind of data-driven. I presume that's what you're doing as well, right, Hari?
Yeah, and we're doing this same. Only the problem comes when somebody, the ethnicity was Hispanic, that tend to be much more admixture population coming from Spain or different portion. That is the one group where the reviewers are giving hard time, and I have no way of answering that question. I'm sure the Asian group is also going to be the same issue. Asia itself is a diverse population. I don't know how we are going to define it as we move forward. So
I see. And so when you say Asian, Hispanic, I presume those are the self-reported descriptors, right?
yes, self-reported descriptor, and then you try to impose on that your genetic ancestry information, which the most reviewers would like. That's when it's not at all compact.
But that's okay. I mean, so we do this all the time. I'm in Miami, and we see, talking about Hispanic, it's like we have Asian, like Brazilians who are Japanese.
Yeah.
If you just ask about ethnicity, you see Hispanic, but when you do ancestry, of course, you're picking up that. And I don't know why your reviewer, I don't know the project that you're doing and presentation, but why the reviewer is having issues with people having multiple identities, and that it's just like we move around.
Sorry. So yeah, I was just going to jump in there. Tony Burnett from EBI. So a few years ago, I was part of the team of the GWAS catalogue at EBI, and we came up with one of these non-perfect ancestry classifiers. There's a publication that I can send it around, but we derived this framework for splitting out self-reported versus some sort of like genotyping, looking for genetic markers in terms of an ancestry classification system. A little while ago, I did a rudimentary analysis of the ancestry information in the data portal for the Human Talent Atlas, data.humantalentatlas.org, which uses the same classification system that GWAS specializes in. And there's some quite interesting results in terms of how the ancestry classifiers map out the Human Talent Atlas community is doing much better than the GWAS community. Great. These systems are not perfect, right? And they do pick up all these kind of cultural biases as soon as you get to self-reported. But there are some workable systems out there that have been published and sort of are acceptable across a lot of the data resources, and leveraging leveraging one of those existing systems, trying to be consistent with it. I think here it's our best chance of getting something that's more likely to be acceptable to reviewers. I can send it to me.
Jasmine?
Jasmine Summer. So Shyam, I think that this is a continuous thread. So when we go to number 3, I think that the gene.
Can I type something about the ancestry point? I'm on Zoom.
Yes, please go ahead.
Hi, my name is Samira Askari. I'm an assistant professor at Icahn School of Medicine at Mount Sinai. I do not know about your reviewer, but I've been both answering to reviewers' comments and reviewing these kind of papers for a few years now. And I think something to keep in mind when writing papers about genetic diversity and ancestry is defining what you mean by genetic ancestry in general for cell atlas or in general. So defining what you mean and laying out the limitations, because there is no perfect way to define ancestry, genetic or otherwise. So defining what you mean, defining its limitations, and then interpreting results within those limitations, basically not overclaiming. In our case has always helped. In terms of, I did not hear all the comments because the audio quality is not good. There is also ways of thinking about ancestry, genetic ancestry in terms of clusters or categories or quantitative as defined by admixture and other methods. And while clustering people in groups have certain advantages in terms of many analysis, maybe thinking about ancestry as a spectrum and a quantitative trait rather than separate groups help both in terms of statistical analysis and getting over some of the issues that reviewers may bring.
Thank you, Samira. Yeah, the audio is much clearer for you than for the microphones in the room. That helps a lot, and I know you have a lot of experience in this. It's a thorny issue. Okay, so I'm sure others have more experience. I've been in a few discussions of how to define or classify ancestry too. The problem is this is one of those topics where everyone has a strong opinion and there's not a lot of agreement. And yet we have to address this issue because we have to move forward. We have to do something, as Samira said, right? Everything we do will be imperfect and flawed, but doing nothing is not a choice either, right? It's not an option. So at least I can lay out what our rough plan, right, of what we plan to do in the Human Diversity Atlas, and then you can all comment on whether we should change it, which is there will be, some self-described ancestry categories or self-reported. It's not easy to use them on a global scale. So as Hari, you're seeing, even in the US, you say Asian, Hispanic, etc., and there's a lot of debate on how useful it is, what it means. And that's just in the US. Those terms don't even mean the same thing in other parts of the world, right? So it's easy to say, oh, we'll use self-reported descriptors. There is no well accepted or universally applicable ontology of self-reported ancestry descriptors that you can use in the whole world, right? Asian doesn't mean the same thing in Asia that it means in the US, for example. So I would say in the HCA GDN, if we're doing integrative analysis across the whole world, those self-reported labels have very limited utility. What are we left with then? What we're left with is a data-driven descriptors, okay? And if you use a data-driven descriptors, the simplest and most obvious thing is just genotype PCAs. And then we can either cluster people based on their genotypes, or we can simply leave them as continuous variables, okay? So just the top n PCs, top n principal components in genotype space. And so each person is a vector of n coordinates in that n-dimensional genotype space. We can use both approaches. Slightly less controversial. I mean, geneticists may say it has limitations technically this way or that way, but at least, and let's face it, it has less political repercussions than any specific ancestry labels we may use, right? So that's we think our path to getting something done in terms of describing ancestry, genetic ancestry, and open to comments on that. If you can all get on board with that, or you feel it won't work, or it's not good, or whatever.
Actually, it's Jasmine again. So I agree with the data-driven descriptors, but also a fusion of the idea of as much meta data as possible, right? So you can include self-reported, and we can see where I always believe in the data driving kind of our interpretation anyway, just from the very primitive genetics. So is it that we also make a call that you have genotyping, that you're not doing right? How close to the ground truth do you want to be? Are you going to assess it through the RNA-seq data? Or are we going to make it so that we prioritize samples that have genotyping across like the new global SNP array? Or is it like whatever you have, you just put it? Because I think that will help us as we move forward collecting more data sets, is to establish a standard right now of kind of the ground truth will be DNA-seq paired with this single cell RNA-seq, but even the type of genotyping array you're putting it on, right?
That's a great question.
Yeah.
Yeah, just quickly, in the GDN, we're really prioritizing data sets that have independent or single cell driven genotyping information. So base level, if you have data from a genotyping chip, a whole exome sequencing, a whole shallow whole genome sequencing or whatever, then at least within your cohort, you can, describe in genotype PCA space, great. And then we'll somehow handle the complication of your genotyping chip is different from someone else's genotyping chip. Rely on the geneticist to help us with that. So that's one level, right? Then there will be data sets where the genotyping information is not available. There's no exome, there's no whole genome, there's no genotyping chip. In those cases, there are methods being developed, and I know Drew Neven is here from Joseph Powell's group at Garvin. So they are developing one method for taking single cell RNA-seq data and inferring genotypes, at least at some level of resolution enough for putting people in some genotype PCA space. Ken Chen has a paper that just got accepted that can, again, infer genotypes from single cell RNA-seq data. So that's our plan B. But plan A is to have some independent genotyping data from the DNA.
So in any case, we can't expect that all data sets will be uniform. Population descriptors are not good, as many here, and Shyam has also said. But what one could do is, if you have whole genome or whole exome data, from which you get genotype data at a reasonable depth, or not depth, I mean, at a reasonable expanse across the genome, there are now statistical, not now, it's been there for a decade or so, methods to estimate the number of ancestors, hypothetical ancestors, and for each population that you have, like for example, from a particular location or a population, with respect to a population descriptor, you can get quantitative information as to what proportion of these of the genome in that particular population is being contributed by these hypothetical ancestors. What one could do, and we are discussing this in a subset of this community, is to use those as quantitative markers of that particular population, the extent of the genome contributed by the P number of hypothetical ancestors, use that as a quantitative variable and then relate it to the single cell data. That's another way of thinking, but I guess the current thought is not to use population descriptors, but use some other kinds of methods by which to, not classify people, but in some ways, what would I say, identify people with multiple ancestors. Patricia has a question.
Yeah, so I'm Patricia from Brazil. Hi, Cheyenne and everyone. I was going to say something similar to what Yasmin just said. But my point more in the sense that there is also no consensus as if we should use DNA arrays or low pass genome analysis, and there are very strong opinions also on those technologies, and some people have generated lots of data on either side and they're not willing to change. And then my question on that would be, how do you suggest, or has this been discussed, how do you want this kind of information to be made ready for human plus analysis, because we are not allowed to share SNP array data or GWAS data is protected, right? So it's not freely available for us to be making it widespread there, like some kind of code, I don't know, like even if we're using different technologies, are you discussing how to do that?
Not even anonymized?
The whole data, no. It's always protected data sets. I mean, maybe having a working group like to discuss that, how to come up with a solution.
I think that some of that was partially discussed in the ethics working group, although not at the depth of specific data types.
I think more like how to make it available.
Yes,
Like because you could
for
have some
collab.
kind of
Yes.
code, but then would we create a code for that?
And who's gonna be the honest broker?
No, so the ethics group, working group that met earlier, actually is developing a working document, or more than a working document, it seems, in terms of data brokerage, HCA, deposition of data, protected data versus data that can be made public. And so there is going to be another component to the HCA that's going to help us navigate those kinds of conversations.
Because
Yes, you're here.
Oh, yeah, sure. Yeah. So, however, my question, yes.
Let me get your mic. Otherwise, even Shyam can't hear you.
So Emily, I coordinate the ethics work, and we did have this discussion. So, so the background is that there will be some controlled access for data that can't go into full open access. So, but the idea was that it would be a whole data set, right? So all the data pertaining to one.
Yeah, but it's just that our ethics don't allow that, right? The ethics
Not even controlled.
Controlled, but not like it's something that belongs to the HCA. It has to be like
Another environment.
because that would be classified as some kind of private bank that we would be sharing the information with.
It's like resharing it like we discussed, if you think about it, because it already exists somewhere.
Somewhere. Okay.
Like, yeah.
That's why I'm thinking of somehow like we should start
Like
to
working on the
discuss
cloud.
how would we code for the, I don't know, but
So in terms of, you mean, when you say coding, in terms of proportion of ancestry, so if you were to look at like
Percentage.
K-12, okay,
Doing the joint
then you
analysis.
would have to say, okay, we're going to look at 12 populations within the Brazilian population, 12
yeah,
subpopulations. and
or
then
the
you have
markers
the-
themselves,
Right.
the SNP markers
Right.
that we use.
So you give us a proportionality like 0.1%, 2%
Yeah.
of Yoruba versus
Yeah, we would have
Western
like
European.
I think that would be, for me, that would be the
That is the way that you'd be able
viable
to.
solution. And that those become variables, when we were
Right.
analyzing the data. So I mean, it has to be built somehow.
So then in that type of data, you wouldn't be able to do EQTL analysis. But it could be used for other things, I guess. I can.
Is this a problem for existing data? Is it less of? Is this a problem than it is for existing cohorts of data? Like, is it something that we should consider addressing going forward? The HCA has always been self-focused only. This group has largely thought about the genetic data, and perhaps we should suggest that going forward, people think about both jointly and the ability to submit that data to the HCA, because it is going to be complicated with these things.
That already exists that they don't maybe have germline DNA actually.
Yeah.
Depends on the institution by how you are learning.
No, but
So we don't necessarily need whole genotype.
you want to identify yourself?
Sorry, Kayla Barnes, Malawi Wellcome Trust. So what about HLA typing? Can we not think about kind of more targeted things that would get away from a whole genome? You know, you're going to have this problem from many countries, especially countries that have had historical kind of not the best historical record with research. So that's most of Africa, Brazil. I mean, it's a lot. So I think this is a pretty important topic. I don't know how the Sangers dealt with that, but there's obviously been a lot of press around how we've used human genomes. So HLA typing is one option. You can derive those from your arrays if you already have them, or you could do some more, well, you can't fully, but you could do some more nuanced HLA typing. It's not that expensive. So there's some other ways around it. And then I guess kind of what we finally got to is you don't necessarily need all the information from the array. We could just pick some key genes that we could use to differentiate. But I agree, if there's just a way as the personal researcher, we could, is it an honor system that, you know, me as my data set, I can say, okay, I'm confident that this is a Malawian from Malawi with this. You don't think that this can be on an honor system?
No, because I think we're trying to, for good or bad, trying to maybe zoom in beyond like country level, because then you can imagine if someone says, well, this is from the USA, okay, right? So, but in Malawi,
Well,
there
no,
are
sorry,
multiple tribes.
I'm not saying this is a Malawian because they've told me they're Malawian. I'm saying they're Malawian because I've looked at my data. Like I've looked at my whole genome data. I'm not going to give
that
you
would
it.
be a-
So actually, if I can jump in, just want to leave
Go ahead.
room for the remaining topics also. This is really a key one. The reason I wanted to try to summarize here is that what we found on a small scale within our own consortium for this genetic data sharing issues and so on, is that it very rapidly gets particular and specific to each team's IRB approvals and national regulations and so on. It's hard to find a global solution for everyone. So I think we will, the way to move forward here is to appoint a representative from each team who can get involved in more detailed discussions with the GDN of what really is allowed or not allowed in terms of data sharing for their network. And then how much can we do, right? And just some simple scenarios. Let's say you cannot under any circumstances share genotype data, full stop, right? Does that mean you're ruled out? No, you're not ruled out. You can still do your, you can download the code or use shared code, do some genetics analysis yourself and upload summary statistics or some other, correlation with covariates or some thing like that, right? Or you can put people in some genotype space, in your own genotype space, which may not be the same as someone else's genotype space, but it's still useful, right? So what I'm trying to say is the solution for addressing each person's data sharing restrictions will be different in each case. It'll depend on the demographics of that cohort, the size of the cohort, the scientific question you're asking, the legal issues, and so on. All I'll say right now is that please appoint someone to talk to us from your team. And we'll have to go back and forth a lot on that.
Sorry, just one other.
Sure.
Just add to that,
Just one other quick comment. Can I just say one other quick comment and then I will relinquish the remote or whatever the, there's two documents right now and that's a little bit confusing. So I think there was an original diversity document and now there's a document that's the diversity 1.0. And I think a lot of people that were on the original document that actually have full data sets haven't been integrated to the 1.0. And like, for example, I put my stuff on 1.0, but then it was edited to something else. So I think those two spreadsheets might need to be married. It's a bit confusing.
Yeah, please email us if we drop the ball along the way. Sorry about that. Yeah, well, we'll set it right.
So I
Samira had something. Yeah.
quick follow-up on what Shyam was saying that yes, in the GWAS world, people go for big data, diverse data sets that they usually cannot share the genotype level data, like none of the cohorts can share. That's what we do, a lot of other groups do. Usually, I think one universal solution, it's not perfect, but it's easy, is giving everybody a set of references. It can be 1,000 genome, it can be many other things, and say, we do PCA, we take PCA loadings, basically, we give you these scripts, run these scripts, give us the PCA loadings. And this gives you for PCA at least one solution that is relatively easy and can be applied to pretty much any level of genetic data. And then for the QTL, I think if everybody follows again some shared instructions, then meta-analysis can be the solution. Do your QTL using this set of instructions and this covariates, and then give us the solution. And then, of course, there will be post-QC to remove any outlier. But I think the one key maybe to all that would be having a group of people who know this and who agree to work on the scripts and then share them with everybody. Because I think not every group have the same level of expertise when it comes necessarily to genetic and diversity and population genetics.
Thank you, Samira. Those are two very practical suggestions. And indeed, for QTLs, we're just sharing summary statistics across cohorts without sharing the genotypes and then doing meta-analyses. And everybody putting their donors into the same PCA space is also eminently practical solution. I want to say, yeah, sorry, go ahead, Partha.
Yeah, Shyam, I think we need to move on, otherwise we'll run out of time and not even have
address the other issues like you just pointed out. All that I would like to emphasize is that we are trying to be embracive of all of humanity, of all of the researchers who are interested in this domain. And we recognize that not everybody can share uniform data sets. And there are ethical challenges. You may not have generated that kind of data, even if you have generated it to share. Please write to us, and let's understand the heterogeneity of the possibilities among the researchers. We can't promise that we'll come up with solutions, but we will try to embrace as many people as possible, because that would actually be the success of the Human Cell Atlas, to be able to draw as broad inferences and as deep inferences as are practicable. So I think I would like to move on from this discussion on ancestry and what is doable and what's not doable to the other questions. Sophia.
So I think we kind of covered 3 as part of the discussion. But what about the methodology component? So not everybody in here, we're not all practitioners of this type of work. So how to associate, what do we do with the data, right? How do we associate genetic variation and donor sample metadata with single cell phenotypes?
Sophia, can we also talk about point 1 about which data sets are ready for inclusion? Because I think someone...
Oh, we moved past that one.
Yeah. So because that's
okay.
a question that just came up, right? Why was my data set in the original spreadsheet, but not in the V1.0 spreadsheet?
Yes.
It's a good question. If I can address that quickly.
So go ahead.
Yeah. So in V1.0, it's a very humble goal of primarily using PBMCs to characterize human diversity. So if you have a PBMC data set that's large enough and good quality, and it's not in that V1.0 spreadsheet, then it should be, and please contact us. If you have a non-PBMC data set, it's quite likely that your non-PBMC data set is in the original GDN spreadsheet waiting to be adopted for integrative analysis, but not quite in the first wave of analysis yet because it's not a PBMC data set. So we're just trying to keep it simple and go one step at a time. We're not ruling out the possibility of including, say, tendon or muscle or something in our very first GDN analysis, but it's not the first thing we will do. I hope that clarifies a little bit. But even if what we're doing violates even this principle I just laid out, please email us. We definitely want more data sets than we have in that spreadsheet right now, for sure. And I will make one more point. It's called Ancestry Networks and it's called Genetic Diversity Network. But we're also we're looking at other dimensions of human diversity. We're looking at sex, male, female, and you can say that's a genetic thing, fine. But yeah, it's not the genetics as in QTLs or whatever. Right? So we will look at sex differences, and we're looking at age differences and environment. Environment is the hardest part. So low-hanging fruit is sex, age, and genetics.
And environment, when we think about environment, also it's within country and also nativities where people were born, where if we have that type of data where people are currently and the time that the sample was collected, where they were. Again, that's dependence of metadata availability and the type of data that we're capturing. I think last year there was a lot of discussion when the GDN members met to decide and describe on the types of metadata that we ought to collect. I don't know how much it was disseminated across all bio networks, but maybe that's something that we have to do again. So that as people are moving forward, Shyam, that we ensure that they're trying their best to collect other types of metadata beyond sex and age. So we capture that type of rich diversity that we would like to take into consideration in other future analyses.
Thank you so much, Sophia. Yeah, so the metadata is key. We don't have time to go over what metadata fields we need in this meeting. So again, I'll go back to email us, set up a contact person on your side, and we'll go back and forth on what metadata we would like to have. But the more metadata we have, the more we can understand the effects of time of day and season and diet and all these other things.
We have a few more minutes left. Is there? Yes, go ahead, Patricia.
I just wanted to make a point.
I just wanted to hear Shyam on the number 2 on methodology, because we do have this issue of collecting samples from different communities by different groups, and then afterwards running the analysis, and then we have confounding variables there, and we don't really know how to go about that. How do you feel, or what do you think about it?
I guess you're asking about the batch effects, point number 2 down below, right?
Yes.
Yeah, it's a fantastic question. Thank you. What was that, Patricia? I can't make out.
Yes, that was Patricia.
Yes, that was, recognize your voice. Okay, so we have seen this substantial batch effects, lab specific, and it's collection specific batch effect. So even if you collect in 5 different parts of the country and process in your own lab, you'll still see the batch effect, most likely, right? So then what do you do? So there are a few half measures or compensations you can do. So one is you say, okay, I'll analyze male-female differences in all 5 parts of the country. So then I'm using internal controls, right? The male-female differentially expressed genes in each part of the country is, internally controlled for the batch effect of collection there, right? Or you'll do QTL analysis in each part of the country and meta-analyze it, or you'll say collection site is a batch effect, right? And treat that as a covariate in your QTL analysis. So these are, so that if I can summarize 2 major strategies. One is internal controls, compare samples from, say, the northern part of the country to other samples from the northern part of the country, right? The other thing is to say part of the country is a covariate that you regress out in any analysis. Those are the 2 major strategies. Batch correction algorithms are great. You can use them for cell typing, but what you're interested in is the biological differences between the different parts of that country, right? And then the batch correction algorithm is the wrong thing to use because it's going to collapse those interesting biological differences. So that's the unique thing about GDN analysis that's different from all the other networks, is that batch correction is dangerous because it removes the biological differences that you're interested in, and your batch and biology are confounded. People from different parts of your country are different batches. They're also different biology. You just have to analyze them separately. So I don't know if that's clear, but each scientific question there has to be handled differently and mostly without batch correction. The only place we use batch correction here is to identify cell types. Or data integration. Yeah. So sorry, I cut you off.
It's okay. Yes, go ahead.
Is there a metadata document we should be working with?
Yes, so I'm thinking like there is a document living in Google somewhere that a year ago, and I think also Cell by Gene and the Ancestry network through CZI has also determined some minimal variables that ought to be collected. Now, how that communicates with the HCA broader network, I'm not sure. You want to talk about it?
Jason Hilton, I'm the Lattice project lead. We do curation for Cell by Gene. We have about 10 or 12 required fields: donor, sample, an assay, and suspension type field. I think is as far as we go. Technical, that's required. We can take any additional fields. And so if this network, any of the bio networks decide on additional fields, we are more than happy to be in that conversation and to help determine what good standards there might be. And then everything will come through us as it gets submitted to Cell by Gene. We can kind of do a double check to see that, everything's in line with the additional standards. Minimal, but there's plenty of room for growth depending on what this group needs.
Can I just make a point?
I just would like to point out that if in the beginning of the project, people don't know what they should be collecting,
Mhm.
then they don't get approvals for that. So we would need better communication so that we would know that beforehand.
Not the one funding project, but that's a conversation that you all have to have, right?
Sophia, there's a question here.
That's a great question. We do have a metadata fields list. I think Kian Hong drew it up, and maybe I think Kian Hong is in the audience there. Kian Hong, you want to introduce yourself and say something about it, and maybe you can email that list to people.
Oh yeah. Hi, I'm Kian Hong. I'm Shyam's dad. So the first thing is that you saw the QR code for GDN roadmap. So that was based on a lot of the discussions we had last year as a network. So over there, I think we have a list of metadata items that was discussed by the network, and I think that's a good starting point. Beyond the atlasing efforts, I think one of the things that we proposed as part of wave 2 of the integration efforts was to have a set of common metadata standards. So I think one of the things that we've been discussing was setting up a working group to try to have or define a common set of metadata and prioritize it according to tiers. Though I think this is something that we're going to have to discuss with Chloe and Lattice and the other metadata teams. So if this is something that you'd be interested in, please reach out. I think they're also defining tier 1 and tier 2 metadata. John, do you want to say something?
Yeah, I just a quick addendum to that. Yeah, I mean, HCA has asked each biological network to come up with that. The tier 2 metadata, which essentially is the super metadata field for its network. We've asked for that information by the end of October. So that's sort of tasks that get to each network, including this one.
Hi, I'm Tammy. I'm a graduate student at the Bird Institute. I have a naive question, and it seems like you're asking people when they're already doing kind of their studies to then help you gather data, PBMCs or data sets from diverse genetic ancestry. And I was curious if there's a more specific effort in just gathering PBMCs for like genetic diversity, or you're initially kind of relying on the community that are already doing this.
So both. The CCI has funded the Ancestral network. And it's the purpose is to capture data, not just PBMCs, but different types of tissue types, cells from diverse populations that are underrepresented currently. And then, of course, you have the HCA that exists already in people in places wherever they are who have a diverse, meaning more than one group that it's already existing. And so we're asking for that type of data also. How many can we have?
You have till 4. 10.
I think we covered a lot of ground. So, and it's really in the audience, and I'm not gonna, I just said your name. And you did a really nice job this morning talking about details and what we can do. And so, of course, that's one of the things, I mean, that's what we kind of started talking about in terms of genetic diversity and what we can do with the data that we're going to be capturing. Shyam has also presented in the past about using single cell seq data and looking at differences across different populations in his cohort. And so the question still persists, I guess. What are we going to do when we collect all this data, and what are the questions that we're thinking about asking? Shyam, you wanna take that on?
Which question? So I tried to lay out
Point
the question.
number 1
Point number 1
of the methodology.
Oh, QTLs. I see. Yeah, so I think point number 1 was about, how single cell QTL analysis is different from your GTEx bulk QTL analysis. At one level, it's not different. So the simplest thing to do is just to take a calculate a pseudobulk transcriptome of each cell type by averaging the transcriptomes of all the cells in that cell type. And certainly that's where we are starting. But then the second level is, the kind of QTL analysis you can do with single cell data that you can't do with bulk data. And there, I think it's really, I don't think there's a well-defined answer. So essentially, people are using single cell data to define a new covariate. Okay, your covariate may be simply principal component 1 in gene expression space. So then you say, you know, as your cell moves from here to here along principal component 1, does the effect size of your QTL change in a continuous manner? So you take that as one more covariate, and then you look for interaction terms between PC1 and genotype, interaction terms between PC1 and sex and genotype, three-way interaction, things like that. So once you add it as a covariate, you can do all kinds of first order, second order, third order interaction terms and so on. Right? So then the question is are the PCs, are gene expression PCs the best way to incorporate continuous single cell phenotypes in your expression QTL analysis? Or instead of PCs, I think there's a paper from Shomo, Soumya Raichaudhry's group, using, if I remember correctly, CCs, so canonical coordinates as the covariates. Then other people have defined some kind of regions in gene expression space and distance from that as a covariate. And I don't know if there's a single good answer here. But those are some of the methods we're trying out.
Any, so I have, so this is all in the context of normal, right?
Or healthy.
Healthy.
Which,
well, yes, healthy.
Non-disease.
Non-disease
But
individuals.
what does that mean? I mean,
that's why I said normal, yeah, with the quotation marks. I can talk about a little bit about the work that I do when I think about normal and healthy. I study hereditary breast and ovarian cancer. So a lot of the people that are in my cohorts are people with BRCA1 and 2 mutations who do not have cancer. And so when I think about this, asking when we do, so we're not genotyping individuals to determine their genetic predisposition to disease, correct, in general. But for those who can share the information, are we able to infer that? Yes.
One way to mitigate this is if you have any longitudinal data of the same individual. So for instance, so my recruitment is children under the age of 1, multiple time points looking at immunity. At 6 weeks before they've even had vaccines in Malawi, 60% of them have a viral, bacterial, or parasite infection. It doesn't mean they're clinically ill, but they physically have a bug in them at the time of collection. So we already know that 2/3 of these kids are going to have a very different immune response. That coupled with the fact that only 40% of them seroconvert to rotavirus vaccine. So then you have another confounder of just overall. So in some of these populations, we may want to think about having some longitudinal data that shows, is this a true signature of diversity or is this a signature of environment? And I know it is one of the kind of metadata points, but I think it's going to be a very tricky thing to get around.
I think that's Kayla, if I'm not mistaken.
Yeah.
Oh, okay. Kayla, thanks for that. That really speaks to the importance of gathering as much metadata as you can. And I've spoken a lot. So Sarah Tishkoff has talked about this as well. So she has a large project in another region where actually, so there's a lot of parasitic infection, and apparently like more than half of her sample collections from people who have a chronic parasitic infection, whose effects you can see in the immune response, right? So what can you do? Get as much phenotypic information as you can and say, treat it as a covariate. In some cases, you can't even do that. These samples are collected in resource poor environments where there's just no medical information, nothing, right? It's still useful as a snapshot of the general trend in that population. So there's something we can extract still. But separating out these kind of gene versus environment differences is a very, very challenging issue. So you've given one example of infants with various infections. Another example, perhaps, just as common, maybe more common, is different ancestries have different diets very often. Even sitting in San Francisco or New York or something, people with different ancestries will have different diets. Certainly in Singapore, people with different ancestries have different diets on average. So we say that you have a gene expression difference between those ancestries. Is it because of the genetics or is it because of the diet? It's not obvious, right? All we can do is describe it, collect as much metadata as possible, right?
Let me make one just quick comment. We can collect more and more metadata, more and more data, more and more covariates, but then sample size will pitch it. You to be able to understand, impact of each covariate and the interactions, we'll need huge sample sizes. So we all recognize that at this point of time, we're just getting an approximate answer to the heterogeneity and probably some important covariates. We can't hope to be able to dissect all covariates because it's only over a period of time that we'll get impact of more and more covariates and interactions between covariates. This is what my feeling is. Shyam, Patty has a question.
Yeah, I was going to say something exactly like that. How granular you get, because if you get too granular, then you don't have, you just get to the individual. And in our project, we had to not use anymore the word healthy because that was not accepted. So we had to have a list of diseases that were not diagnosed because even maybe they even have it, but they don't know, right? So we had to change everything to not having this list of disease diagnosed, but that's all we could do.
I think we can only get 2 more questions and comments.
Just on that note, though, it's sample numbers is the game, right? So we can collect as much metadata, but we just need to deposit it. You can't get at QTLs the more you stratify. So I would say, even with the BRCA stuff, it's like we don't know they're going to get disease. We don't know that genotype is going to drive them to disease. We're going to treat everybody as quote unquote normal and deposit data. That's how GWAS did it and expand it through Ancestry. We can't keep every covariable or we won't find anything. But the more we deposit and the more we emphasize that in the community and the more we build out these, we really need an Excel file that just says these are the metadata that we would like to collect. And over time, we keep that metric and then we expand on it. But maybe it's just a regional effect where there's parasitic infection that's driving that, but we won't know until we deposit all the data sets. So I would say we need to establish what that Excel file looks like, what the headers are, but then just deposit data so that we can really establish, and genotype, right? So that we can just establish by, it could be a regional thing, a disease thing, et cetera, but we won't know until we just put numbers and numbers and numbers in.
Great suggestion. From our side, we promise we'll at least get that list of metadata to JSON so that when you're submitting to Lattice and you say you want it included in the genetic diversity network, it'll be part of the submission process. In addition, we will make that spreadsheet of metadata fields available on the mailing list of the genetic diversity network. If you're not in the genetic diversity network mailing list, again, please email me or anyone else in this panel.
So one last question.
Last question. This is about the longitudinal. We have more than 200 women who donated their tissue 5 years apart. These are clinically healthy women. That's the terminology I need to use. So we did spatial transcriptomics on these paired samples. The problem with the data analysis is that few women, the BMI is completely different. Some others, BMI did not change in 5 years. So the data interpretation as a matter of age or something becomes so complicated. The data we have already submitted to Lattice. So hopefully the sample size is small because spatial transcriptome is too expensive to do it. And we did about 5 pair of samples, only data we got from 3 of them. So, it's expensive, but that's where things field has to go next.
So thank you everybody for your comments. Lots of great conversation points. We have a note taker who is summarizing all the points, salient points. We have some action items to distribute on the metadata Excel spreadsheet requirements to the GDN members, and hopefully those who are not part of the GDN can join and get access to that information. It's not hidden. And And please fill out the Google 2 tabs. One, if you have data sets that you're willing to share or can share. And then we also have to discuss how to share for those who cannot share, what types of data can be shared, or at least computationally, how things can be done. So we're using the same tools to ask the same questions across different data sets. Yeah. Thank you very much, everybody.
Thank you, guys.